Hello again, and we are going to be moving on to discussing these enzyme and protein functions in the DNA replication process. So we're going to start here with something called helicase. Again, this is an enzyme, as you can see, ACE, that unwinds the double helix, as shown here in green, and pulls apart those nitrogenous bases so that we can have access to the template. Now another protein shown here is the single strand binding protein, and those are shown here in gray, and again those are attaching to the template so that the two strands cannot come back together during this replication process. We want to keep them separate so that we can get those enzymes in there to add the new strands, or the nucleotides of the new strands. So that's true for both the, of course, leading and lagging strand. And then the next enzyme to come into play would actually be RNA primase. I know I have them listed kind of in opposite order here. And it's to prove a point, or to show a point, that this enzyme DNA polymerase 3, which is really the star of the show, I'd have to say, comes in and adds the new nucleotides, but it can only add them to the 3' end. And remember that 3' end has that hydroxyl group, and that hydroxyl group is needed to catalyze the reaction. Okay? And the thing about DNA polymerase 3 is that it cannot just synthesize a strand without having a 3' end of a nucleotide strand to add on to. It needs to have something to grab onto. It needs a 3' end of a nucleotide to add these new nucleotides that are coming in. So that's where our RNA primase comes in, and it adds a short sequence of RNA. And in that RNA is very similar to DNA, as you know. It has a 3' end, and the RNA, or the DNA polymerase, can then add the new nucleotides to that free 3' end with that wonderful hydroxyl group that's necessary. All right, so we will look at the leading versus the lagging. The leading strand synthesis is pretty straightforward. The process begins with adding the primer with that primase. Of course, we've used the helicase and the single-stranded binding proteins, which are not shown here, just to simplify the picture. But then the primase comes in. We add a short sequence of RNA indicated by this red square rectangle, and then our DNA polymerase 3 starts adding these new nucleotides. Remember, here's our 3' end. Let's look at the strandedness really quick. 3' to 5'. So this strand must be running 5' to 3'. So this would be the 3' end of this little tiny piece of RNA. So our DNA polymerase 3 can add nucleotides to the 3' end. Perfect. It adds the nucleotides, no problem. It just swings along, keeps adding them. And that's pretty much the case for the leading strand. Now at the very end of the process, of course, we have to change out all of these primers. They're RNA, remember, with DNA. And that's where the DNA polymerase 1 comes in, and it'll change out the RNA for DNA. And then between this little primer and the new DNA nucleotides, we have a little gap. So we need DNA ligase to ligate the backbone together. And honestly, that really is it for the leading strand. It's a pretty straightforward process. Now, the lagging strand utilizes all of the same enzymes and proteins. However, it does it in a little different fashion. It's a little bit more complex. So, as you can see here, we have our lagging strand, and we're going to jump over to this slide, which really shows us the synthesis, a little bit more detailed process of the synthesis of this. It's just showing us this lagging strand here. It's not showing us the other strand, just for simplicity. So we have our list of enzymes. They're all the same. The helicase and the single-stranded binding proteins, those have done their thing, as you can say. And then the primase. So here we have our primase put down. Here's another primase. That little pink guy is the primase putting down the primer. And being as though, let's go back here, this is the 3' end of the molecule, and that is the 5' end of the molecule. This would, of course, be the 3' end of the new molecule, the new DNA. This would be the 5' end. So we can't add nucleotides to the 5' end and start to go toward the fork. We have to add them to the 3' end. So this would be the 3' end of this little primer. So the DNA polymerase 3 can only add nucleotides to this end right here. So it would have to kind of add them and then move this way until it hits a primer, and then that one will fall off, and then it'll jump up here, and we'll go through this process kind of over and over again. So one more time, this is the 3' end of the molecule. Over here, that's the 5' end. So this new primer, okay, this would be the, again, 3', that's the 5'. Okay, so that would be the 3' end of the molecule, this would be the 5' end. And you can't add nucleotides to that 5' end. So again, the DNA polymerase 3 jumps on, adds nucleotides until it hits a primer. It'll fall off as indicated here. And then what will happen is a primer, which is not shown, will be added here. DNA polymerase 3 will jump on, add nucleotides, again 3' to 5', and then fall off again. And the process repeats and repeats and repeats. So it's kind of like taking 2 steps forward and 1 step back, as you can say, as we're doing this lagging strand. So our overall direction needs to be toward the fork, which the fork is over here. Remember, the fork is here. But what's going on is the DNA polymerase 3 has to kind of work backwards and then jump forward. It works backwards and then jumps forward. So it takes a lot longer to synthesize this strand. All right, and then we have our DNA polymerase 1 shown here in yellow. It's taking out those RNA nucleotides and replacing them with DNA. And then finally, we have our DNA ligase, which plays a much bigger role here in the lagging strand because there are so many of these little fragments that are formed. These little fragments that are formed between the primers are called Okazaki fragments. And they are named that after the wonderful Japanese scientist who detailed this process, and he named these fragments after himself. So DNA ligase, again, we're going to ligate together the backbone of the DNA. All right. So this last portion of the video will talk about just DNA packaging. So you're going to want to know the 4 levels of packaging. Level 1 would just be the DNA wound around those histones. Make sure you understand the charges. DNA is negative overall. The histones are overall positive. That's why they are able to stick together. And it kind of looks like beads on a string. So know the sizes and the common names for these levels of packaging. And then we have level 2, which is kind of like taking beads on a string and wrapping it around a central axis. This is a horrible picture showing this conformation. But again, this second level of packaging, then we're just getting more and more condensed to fit the DNA in the nucleus. And so for level 1, it's a 10 nanometer fiber because when you measure the actual thickness of the fiber, it's 10 nanometers. And as we move, getting more and more packaged, we increase the thickness of the strand. So 30 nanometers here in level 2. Level 3, you take that beads on a string wrapped around a central axis, and you attach it to a protein scaffold. So it's not histone protein, but it's another kind of protein. And you attach it in these kind of looped domains. Okay? And then that is a 300 nanometer fiber. And then finally, you fold this strand back on itself, and it is a 700 nanometer fiber. And of course, when you have the 2 sisters together, if you're going through mitosis or meiosis, you'd have a 1400 nanometer fiber if you take both of them together. Okay, so we're going to pick back up with talking about euchromatin, heterochromatin more in a later chapter. So we'll end this right here for now. Hope this helped you understand the DNA replication process.